All right, why don't we get started? I'm very happy to continue our Fireside Chat series. And this particular uh, Fireside Chat is related to a topic that I think is one of the most important ones when it comes to science and trying to make an impact with one's science. So I'm very happy to have Maurizio Vecchione joining us today. So you may have seen that Maurizio recently joined us as the Chief Innovation Officer at the Terasaki Institute. And it's fantastic because he comes with a wealth of experience in the area of innovation. He led and built the whole program with Bill Gates called in an organization called Global Good for the past 8 years. And before that, he has done lots of amazing experience, including being a serial entrepreneur and having done a number of different innovations himself. So Maurizio, welcome to our Fireside Chat.
Thank you so much, Ali. It's a pleasure to be here to talk about these sort of very important topics.
Wonderful. And before we get to Maurizio's introduction and background a little bit more, with him explaining, I just wanted to, for anyone who's new to the series, I wanted to talk a little bit about this, the Institute and what we're all about. This picture on the left-hand side is probably where it first started. This is a picture of the first transplant that occurred back in Boston at the Brigham Hospital, which is a place I used to work at, where Dr. Joseph Murray performed the first kidney transplant. And this transplant changed medicine in many ways, because what it enabled people to do is to be able to take organs and be able to treat a variety of different diseases. Now, the challenge with this approach has been that initially it was really limited to twins, because it was difficult to be able to identify whose organs would match with which other individual. And this problem wasn't really solved until Dr. Terasaki came around. Dr. Terasaki was a professor at UCLA and subsequently a founder of a company that was taking his innovations from what he did research at UCLA and made it into a viable product for this field. And really the concept was in identifying the right HLA matching, this is the right antigens between the donor and the recipient, such that the transplanted organ would survive. So this is really one of the first application of what we call now personalized medicine, where the therapeutic is personalized for the patient. And what we're trying to do at the Institute, which has been endowed by Dr. Terasaki and his family, is to innovate these personalized wellnesses and cures, and not only be able to discover approaches to do this, but be able to foster real applications with these approaches. So we currently have a number of different buildings that are located in the Los Angeles area. Our flagship building will soon be a large building in the Woodland Hills neighborhood in Los Angeles, which is going to enable us to really do top-notch scientific research. And before we start, I do want us to tell you that we're very active in a number of different activities that I think will be of interest to many people. And I do encourage everyone to follow us on our social media platforms, as well as look at our website that's listed below, where we do have a number of these videos of other seminars called Terasaki Talks, as well as a number of other fireside chats. So with that said, I'd like to turn the floor over to Maurizio so he can tell us a little bit about his own background and what got him interested in innovation.
Sure. Well, thank you, Ali, for that introduction. And, you know, some of you probably have heard me talk in other venues about the role of innovation uh, in the world of impact and in the world of impact toward the patient. You know, innovation has been defined many ways, and often it's associated with what I would consider private sector type of activities. So the building of companies, the launching of companies, the startup community, and all of those are, of course, important ingredients in what makes for an innovation program. But at its purest sense, innovation is how science breakthroughs are going to reach the patient. And without a focus on innovation and what I think Ali and I are starting to call an innovation culture, it is very difficult to translate what may be great scientific discoveries or breakthroughs into patient life-saving and patient improving quality of life type of solutions that ultimately solve real problems and can be made accessible to everyone. So building an innovation program at Terasaki, to us, is more than a nice thing to have. To us, is an essential part of how we're going to help realize the vision that I think, Ali, you just expressed, of having some of the possibilities that were kickstarted in many ways by Dr. Terasaki with his innovative work, but that now are really mushrooming into what I would describe as a real revolution in the way medicine is going to be practiced, if not today, in the very near future. And that's going to be a revolution in how people think about disease, in how people think about prevention of disease, in how people recover from disease, and this whole sort of area of wellness. And so, you know, what's exciting is the convergence of multiple sciences. And I think, Ali, in the past, you've talked about convergent science. The convergence of these sciences give us an opportunity to do some truly groundbreaking innovation that ultimately may be groundbreaking in its impact. And that's really what gets me excited. And of course, to sort of go back to your original question, which is a little bit of background, you know, I got to spend the last 8 years working for Bill Gates in the context of some of humanity's most daunting medical problems, including in the most fragile populations in the world, to really look at what is the role of innovation in solving problems that ultimately have not just millions of people worth of impact, but billions of people worth of impact. And so to me, it's really exciting that science can be a tool to have those kind of impacts. And I want to tell you more about it today on what may be our strategies, as well as sort of the end vision for where some of the innovation that is in pipeline, some of the science that is in pipeline will spawn in terms of innovation.
Wonderful. And I think very well said. You know, one of the interesting things when I first got exposed to science is that my own personal journey took me through the lab of another one of history's pioneers, Bob Langer. And I think that really changed my view of scientific impact and how it can really make a big difference in the world. And I think in many ways, many of us scientists get into science because of curiosity and just the intellectual stimulation. But once we understand the actual impact that science can have, I think it's another level of excitement and stimulation. And I think it's very clear in what you just mentioned. So let's go through this process. And I did wanted to kind of take us to the next slide, which is really, in your opinion, what does translation entail? And maybe you can tell us about what this slide is.
Yeah, absolutely. So this is, and I apologize, it may be hard to read on some screens. It's sort of a classic vision for the journey that scientific innovation on the left becomes products that save lives on the right. And, you know, we're all living in the midst of this pandemic, and I think a lot of us have learned, even the general public, it's now household conversation to talk about, you know, phase 1 and phase 2 and phase 3, and when will the vaccines come and all those kind of things. And you know, politicians are found to say that they're going to accelerate that process, et cetera, et cetera. And you know, there is a tendency to forget that these processes are actually mired into best practices that are actually designed to season and mature the lab experiment that may be on the left side into something that reliably can address human condition. And there are many, many factors that go into this translational journey. Now, this happens to be a diagram that comes out of the FDA, and it's somewhat of a classic view. And you kind of see siluses and sort of hard lines that suggest that you reach a certain stage. The T's at the top is sort of an indication of readiness for the patient. So T0 is still a lab experiment, and T4 is a product that can be safely used. And you know, the journey is really designed to be a de-risking journey. And it's really designed to essentially reduce the risk so that you get to a point where it's not risk-free. Medicine rarely is completely risk-free, but the risks that are still associated with the piece of innovation are small enough that they become acceptable in the context of the advantage or the value that medicine. So if you know, any of you that have used the drug will know, I mean a legal drug, will know that there are counterindications and there are protocols where that drug is delivered. And those really mire from the learnings of this sort of de-risk journey. And the same is true for devices, right? There's magic in modern imaging, for example. There are small risks, radiation if it's X-ray, for example. And so it's really a question of how do we enable physicians, clinicians, and patients to ultimately make those risk-reward decisions? And do we have some unforeseen risks that we want to catch before the general population of patients ends up using it? So this is a necessary process. And what I want you to see is the blue or lightly shaded blue boxes, because those are really the things you have to do during that innovation stage. So on the left side is the actual research side. And obviously there are things to the left of the left that go on. But what I want you to see is that there's a lot of work in those blue boxes. And it's a lot of work in terms of time. It's a lot of work in terms of resources. And there is a lot of risk of failures in those later stages, which means the sort of late stage translational aspect is not something that you can improvise. It's something that you have to be systemic about. You have to be extremely collaborative because it is extremely rare to have just one organization that have everything. And, you know, to take it back to what we're doing at Terasaki, you know, our academic affiliations, our collaborative culture, and the fact that we're an independent foundation means that we have the ability to really build this sort of grand coalition of collaborators that can drive all phases of these things on an accelerated basis. Now we'll talk more about that, but maybe Ali, you want to go to the next slide?
Sure, which really I think is an important slide because it does talk about something I'm very interested in, which is how do we fund this?
Exactly. So, you know, this is a slide that comes out of a different NIH document that is sort of illustrating how this type of work, well, how all biomedical research is funded, but then, you know, what happens in the later stages. And for those of you that are non-scientists and are not sort of on the front line of doing this kind of research and figuring out how to get it funded, the left side of this diagram is really the research stage. And that's when partnerships with the NIH, the NCI, the NSF, all these sort of government acronyms, as well as sometimes other type of funding agencies, as well as international governments and agencies. But basically, the pure research side is the role of government. And it is typically government funding that drives that. In our case, you know, we have the luxury of having an endowment that supports what I would call a core effort. But, you know, I know, Ali, you have, I don't know if you want to give the number, but I know an enormous amount of energy is going into grant applications and effort, and the team has had a tremendous success rate, as well as I think there's acceleration in that. And NIH and NSF are great funders. But there's a limit on what they will fund. And that limit often expresses itself not just in dollars, but in stage of development. Their mission really is to nurture that left side of this equation. And the lines in this graph represent the cost or the cash flow of a biomedical innovation. And so you can see that by the time the line becomes green, it means it's actually generating its own funding. And of course, the classic example could be very much what happened to Dr. Terasaki in your story initially, right? He translated his discoveries into a company. That company became very successful and ultimately created not only his personal wealth and the family's wealth, but his ability to fund more research out of his own endowment, which of course the Terasaki Institute is the legacy of. And that's when everything goes great. But notice there's that sort of dip, deeply red dip, which is representing the cost of all the blue boxes in the prior slide. And if you can see, the cost of those phases, which are also highlighted here in that gray bar, is actually much larger than the cost of the early stages on the left side. Because often that's when you're really facing the full brunt of the clinical validation. That's when you have to go to the patients. That's when you have to enlist the full protocol of the regulatory environment to ultimately prove the safety and prove the efficacy. And of course, the depth of this valley varies with what you're doing. If there's relatively high risk innovation that ends up in the body, that is a more costly type of effort than if you're building a medical device that maybe is an improvement of something that already exists. And so in the parlance becomes something that can be done through abbreviated regulatory process and more de minimis type of research. And this is an evolving picture in the sense that both the regulatory environment and the best practices associated with how you validate these technologies are constantly changing. So as an institute, we have agility, and we have to have agility to sort of figure out what's the best pathway through this. But the fact remains is that this is the expensive part, and this is also when a lot of those government sources sort of stop, which means part of what we have to do in building an innovation culture is figuring out how do we create a partnership culture, whether it be companies and private sector, whether it be capital sources and venture capitalists, whether it be donors, that can jump into supporting that part of the effort. And of course, in our case, it's more of a co-funding arrangement because we have the ability to, of course, put our own money into some of these phases. But this is really a challenge. It's such a challenge that the industry has a word for it. They call this the valley of death, because so many innovations don't ever make it through this sort of dip in funding to the other side, where they actually have a chance to deliver their impact and become thriving, self-sustaining companies. This is also the role of foundations. And so, you know, it's a lot of the space that I work with, you know, with Gates. And Gates, of course, is a tremendous investor in this space. You know, between the Gates Foundation and some of the efforts at Gates Ventures, they're among the largest funders in the world for these kind of activities, but they also have a very precise focus. And so part of our art is going to be to match the vision that we have for the kind of impact we want to deliver with the appetites that our donor community will have for those investments, and of course the appetites of the private sector to sort of help filter. So this is a curation of a collaboration that spans the funding, as well as the science, as well as ultimately the delivery on the right side of this, which typically would mean private industry. And all of this needs to be done in a very careful, balanced way to make sure that we don't lose sight of the impact mission. You know, again, the reason why we want innovation is because without innovation, we can't deliver the impact.
There we go, which leads to this slide.
Exactly. And this is the fundamental equation that moves me, I think moves you, Ali. And I think as we think back about the best practices in the biotech and biomedical field, leads also to some of the greatest success stories. You know, you mentioned Bob Langer. He is clearly one of the biggest success stories on how to do this well. Langer Lab at MIT is a legend in many of these areas. Both you and I had an opportunity to work with Bob in various stages of his effort. And you know, this equation that innovation is the tool to deliver the impact is really what moves us. You know, yes, money could be generated and that could fuel more research and more innovation. Yes, great companies could be created that will have another set of beneficial stakeholders. Yes, you know, there is lots of value to the research side on the left side, and sometimes you need to research things that don't make it all the way through, and there's still a lot of value to that. You know, friends in the biotech industry tell me that learning from their failures is just as important as the successes. And those of you that are tracking the trends in artificial intelligence, for example, for drug design and drug delivery, will know that mining the failures from a lot of the drug companies is actually teaching a computer how to find the next successes. So there is lots of value to doing innovation that doesn't actually make it all the way to impact, but almost without fail, if we're going to deliver an impact with a piece of technology or a piece of science that we've created, it is the innovation component that will actually deliver it to the patient. Okay.
Wonderful. And which takes us to different types of diseases, and how do we actually try to decipher and look into different opportunities?
So this is a busy slide, and I realize most of you won't be able to actually read it, but it has some interesting implication that you can tell from the color. So what this is a slide from an institute that I actually worked very closely with when I was with Gates. It's an institute created as part of the Population Health Center at the University of Washington. And it's often been described as one of the world's leading repository of medical data. And Chris Murray, who leads this effort, has been the guy that invented the concept of the global burden of disease. And essentially this is capturing, on this one slide, is capturing the importance and the associated impact of every disease known to mankind. And the red boxes are infected diseases. And in aggregate, the size of all those red and orange boxes give you a sense of how much of the global burden of disease comes from infected diseases. And the blue boxes are so-called non-communicable diseases. Those are things like cancer and cardiovascular disease, and of course, those of you that are medically trained will probably point out that some cancers are infective diseases and so on. So there's some blurred lines. And the intensity of the colors tell you whether from an epidemiological perspective, the disease is growing, the darker intensity, or whether the disease is shrinking. And this is, of course, done globally. But at HME, they also look at very much the local region. So you could ask the same question about the US, you could ask the same question about you know, the developing world, et cetera, et cetera. It suffices to say that there are a lot of diseases. And from our perspective, one of the things that I think is a key takeaway when you look at this picture is that it's hard to say which single disease moves the needle on human health. And it's hard to say that one disease should be the focal of every effort, because if we only resolved it, we would significantly improve the world. And that statement needs to be weighted on the fact that most funding, especially on the innovation side, is disease specific. So I can show you folks that want to fund cardiovascular disease. I can show you folks that want to fund certain types of cancers. And by the way, in a lot of their funding calls, they take the approach that their disease is the most important. And of course, if you have that disease, it is the most important. But increasingly, the real burden of disease in society is coming from what I would describe as comorbidity. Take even what's going on with COVID. Okay, it's an infective disease. But people actually are not necessarily dying of COVID. They're dying of lower respiratory or cardiovascular complications, and often aggravated by comorbidity. They might have diabetes, they might have some other factors. And so this sort of silencing of disease and looking at programs and solutions that apply to one disease is really becoming increasingly ineffective. You know, at Gates, we had pillars that were very disease-centric. And for example, one of the commitments at Gates was the eradication of an infective disease called polio, which, by the way, Gates is very close to achieving. It's the first time in history, 5,000 years of history, that a disease has been eradicated other than one time before, which was smallpox, okay? So imagine the sum total of human ingenuity and technology and science having taken 5,000 years to get to the point that we can actually eradicate a disease. And that, by the way, is still not achieved. So there's still work to be done. That's fantastic, that's tremendous. But let's look at the context of COVID, for example. COVID is overwhelmingly impacting low and middle income countries because of the obvious nature of the difficulty in social distancing, the sort of relatively poor state of the healthcare system. And so COVID is interfering with the ability that, for example, at Gates existed to continue moving on other diseases. And suddenly at Gates, it was discovered that repurposing some of the infrastructure that was built for polio was actually critical in improving COVID and other diseases. So this idea of interdisciplinary approaches is pervading the disease landscape. It's ultimately pervading the impact landscape. And it clearly plays a role then in how we should approach the science and the innovation, because as an institution that is really good at bringing together interdisciplinary and different approaches, we're sort of ideally suited to create innovations that don't just work in one of these boxes, but work across and spanning across the boxes. And ultimately, it's really about population health.
Yeah, wonderful. And that you mentioned the word interdisciplinary, and I think that kind of nicely segments us to the next slide here. So if you can talk about this, that would be fantastic.
Yeah, so this concept of the interdisciplinarity of disease is illustrated in a COVID sense, just as an example. So this is a chart that shows you all the epidemics, so it's infective disease, that had pandemic potential, and that happened since 1988. Okay, there's a lot of them. So you may think that COVID is a once in 100 years or once in 500 years that was unpredictable. But anybody in population health and global health will tell you that, including, by the way, my ex-boss, Bill had a TED Talk in 2005 where he actually predicted the details of what we're living through with COVID. Okay, now, is he Is he a genius, or does he have a magic crystal ball, or is he just looking at the science? And this is what the science is telling us, that we are causing, or at least participating or complicit in factors that are highly interdisciplinary, that are accelerating the pandemic potential. And actually, Dr. Fauci, just a few days ago, had a public release where he coined a new phrase that we're now living in the age of pandemic. And so, while I don't want to depress anybody, the fact that pandemics are accelerating begs the question of why. Why do we have so many in the last few decades, and why we went hundreds of years before without pandemics? Yeah, really, the pandemics before that was probably the bubonic plague, you know, of the Middle Ages and the Renaissance. And so you had hundreds of years that went by before you started having this acceleration. Well, a lot of people think it's because of this sort of interdisciplinary interaction of what we're doing to the planet and to the planetary health. The WHO issued a very large report looking at how the genetic pressure and looking at how our close infringement in both the animal world as well as from a climate perspective, the changes that the environmental pressures are putting on genetics are driving this acceleration. And of course, there's a lot of undiscovered science, and I don't want to sit here preaching about necessarily pandemics, but the point I'm making is there are multifactorial reasons of how disease is evolving and how the treatments need to evolve and how the diagnostic needs to evolve and how the prevention needs to evolve and how the wellness needs to evolve. And they're all compounded by the fact that as populations, we're growing rapidly. We're heading toward that 11 billion people planet. And these pressures are only going to be exaggerated. And one way in which I think you can begin to look at resolution of these issues is to look at the genomic profile of individuals and to leverage, therefore, what genetics can tell us about the disease and the interaction between the patient and the disease to really optimize the solution. And so this concept of precision medicine, Ali, that you introduced at the beginning, in my opinion, is not just a great thing for the evolution of medicine, but it's an essential thing for how medicine will be practiced in the next few decades. And it's not just, by the way, technology for the rich. This is not just building customized medicine. You know, I'll give you a perfect example. Right now in the poor world, one of the scourges is tuberculosis. And tuberculosis is becoming drug resistant because we have not asked the questions of host response and the questions of personalizing treatments to individuals. And it's now becoming a pandemic potential, or at least an epidemic potential everywhere because of that factor. So we are a village, and precision medicine has a role in that village. And I think the kind of technologies that deliver better, cheaper, faster precision medicine are going to be pivotal in the future of healthcare. I think I just spoke to that.
Yeah, you partially mentioned that and answered that. So maybe we'll go to the next slide after this.
Yeah.
So this is talking about the model. Is there a model for what we're trying to do? And
yeah, let me say that there are definitely models. We mentioned one in Bob Langer and Langer Lab at MIT. A couple of sort of data points, though. There are not a lot of viable models to do this on the West Coast. So and definitely in Southern California. So while there is incredibly vibrant scientific development community, the sort of integration of the sort of blend of interdisciplinary approach focused around multi-disease and working specifically in conjunct with an innovation platform that can accelerate implementations, those kind of things are pretty rare because they're difficult to do in classic academic institutions. what's the sort of defining institution inside a university? It's the department. And the department by definition is single discipline. And while there's a lot of interdisciplinary efforts starting to happen and also translational efforts, having an institute of this size and capabilities of what we're doing here at Terasaki is a bit unique. And so, the model here is to really blend the business disciplines that will ultimately launch and incubate companies, the commercial translation, including the regulatory pieces and the clinical pieces, and the science side, blend them all together to sort of drive toward impact, and do them with an eye on what the final impact is, which means that often we need to start thinking about what is the problem that we're solving beyond the discovery of new science, well early in this sort of translational journey. So maybe, Ali, we can move to the next.
Yes. Oh, sorry, I was on mute. I was going to mention that I know we have a couple examples of the types of technologies that that are relevant to some of the things that we're experts at. So, and you mentioned some of this personalization, and I can maybe give a little bit of insight into some of these and how they're relevant to personalized medicine. But I know bioprinting, for example, and 3D printing in general are very much aligned with the concepts that you mentioned, which is being able to design specific patient-specific kind of therapies, or be able to address challenges with finding better treatments, potentially for emerging diseases like the ones that you mentioned, by being able to make these more functional tissues. So that's kind of one of the examples, but if you have any additional comments on that, love to hear.
No, I think, I sort of asked you to talk about the 3D tissue printing and the bioprinting, because for those of you that are not in the business, so to speak, that's a pretty wild idea. And the idea that you could, 3D print, maybe even 3D print in the body, tissue that has regenerative properties or is part of a regenerative process, is really unbelievable. And just think about the science fiction like possibilities if you carry that forward and you think about what the future of medicine could be, from restoring cartilage in orthopedics applications to literally repairing organs, and that this is not science fiction, though. This is happening in Terasaki today. Okay, now obviously we're not 3D printing a heart, although there was some recent publication where they went a long way to that. Okay, so these are sort of an example of what happens when you bring together these disciplines. Look at what's embodied in this. It's you have the frontiers of material sciences, you have the frontiers of biotechnology, of genomics, you have engineering, the frontiers of engineering along the lines of the chemistry and the manufacturing technologies, the printing technology. And you have a huge component of data science and analytics that all integrate together to provide solutions like this. So I thought this was excellent, but maybe, Ali, a question for you. I also think perhaps a slightly closer to reality and to the patient set of technologies that I think are fascinating is the idea of having biomaterials that are smart materials that can sense and, for example, trigger drug release in ways that can accelerate healing or can have other sort of almost magical property. Do you want to make a mention?
Yeah, definitely.
Of frontiers on the biomaterial side.
Definitely. And I think, as you mentioned, as our understanding of biology enhances, and particularly the biology of tissue development and biology of tissue healing, then we can start thinking about, instead of making the entire tissue, think about how we can recreate regeneration or processes that are important during tissue formation inside the body of a patient. And that's where areas like advanced biomaterials and this whole area of smart or personalized biomaterials becomes important, because we may not need to print the whole tissue, but we may be able to engineer materials that can be placed inside a patient's body that can basically enhance the restoration of the tissue and its function.
Yeah, I think
Wonderful.
that's fascinating. Yeah. And again, all of these things in aggregate have the potential to change a lot of clinical practice. And we talked a lot about surgical technologies, but this also has the potential of delivering what one would describe as theragnostic, the combination of therapeutics and diagnostic. You know, the kind of diagnostic, maybe it's a wearable, maybe it's implanted, that can react to conditions in ways that then it can decide what drug therapy or how to deliver and when to deliver, you know, therapeutic agents. And if you think about, you know, some of the problems surrounding drug delivery, where the right dosage at the right time, I think I saw statistics recently that said something like 38% of treatments that fail in patients is because the patient is not compliant and forgets to take the medicine at the right time, or takes the medicine regardless of whether they need it, etc. So being able to sort of automate those processes and make them intelligent and deliver, again, in a personal way, what you need, when you need it, is also part of the implications of what you're doing, which I think are all groundbreaking.
Yeah, and that I think nicely leads us to the other slide here, which is the applications of these types of approaches for drug discovery. And what you see here, which is one of the things we've previously worked on, is to try to develop models of human physiology outside of the human. So the idea is to be able to take advances in cell sciences and techniques like 3D printing and new materials to be able to build tissues that are tailored, that are basically analogous to a person's own different tissue types, and then be able to interact even different tissues to each other to create a more complex biological systems. And I think this is another area where I think that, will have huge impact in medicine, because it really allows us to decrease the time it takes to develop drugs and make them cheaper to develop. And by doing that, you can potentially even make it more widely available globally as well.
Yeah, and let me amplify that. What most folks in drug discovery speak of when it comes time to validating the mechanism of action of a potential new drug is they speak of animal models. And the reason why they speak of animal models is because they need a physiological system to essentially test out the assumptions that went into engineering the drug. And you know, so setting aside what you feel about the ethics of animal studies, the reality is that they're only partially effective in modeling the real human body, let alone an individual human body. And so if we had this sort of grand vision of having a chip that somehow can contain a realistic, perhaps even personal model of the disease and the body, and you had that chip or a network of those chips behave as a realistic model, you could envision a future where we skip the animal model, we go straight to this kind of personalized testing on a chip using these customized tissues or culture lines or cell lines and can even ask questions around specific types of patients, and validate, even if it's not a new drug, validate the regimen of drugs that would have the highest impact. And this is, of course, an area that is deeply attached to the bioinformatics world and also the artificial intelligence world. And so it really is.
Brings us into this slide.
Yeah, exactly. And this is a slide that comes out of a government research laboratory environment, but it's sort of a vision for the drugs development cycle of the future. And all of the boxes above the green lines are actually in silico. They're actually digital technologies. And there are literally moonshot programs at places like the NCI that are federating some of the largest supercomputers in the world, including the national supercomputing infrastructures, to take the data sets about molecules and their performance and create sort of a first pass at identifying the kind of compounds that should have an effect against a particular target. But that loop is imperfect. There's simply not enough knowledge in the data as well as in the mechanistic understanding of how the chemistry interacts with the physiology to be able to then not do the green loop. And the green loop is essentially integrating this sort of organ on a chip and laboratory on a chip technology so that the computer can manage the sort of testing and create a feedback loop that goes and fine-tunes the artificial intelligence to start building better compounds. And you can see right there in those green boxes is where the technologies we just described fit. And I find it tremendously exciting because I think in the future, developing customized treatments against specific patient targets, and it's not just the patient, but perhaps the class of genetic evolution of their disease. And those of you that track cancer, you know that the frontiers today, you know, there is no such thing as a generic cancer drug that is effective in all situations. A lot of cancer evolves and it evolves through stages, it evolves against other factors, and often even develops a resistance to certain chemotherapy. And a lot of that is because we're kind of using a one-size-fits-all approach. If we could test against specific genetic profiles and tissue profiles, we could essentially create an optimization loop where you're giving, again, the patient what he or she needs, when they need it, and in a way that stays ahead of creating a kind of mutation or genetic evolution that will ultimately create resistance to the treatment. And so this is not just a faster way of developing drug. This is not just about operationally improving the drug development cycle and the efficacy, but this is really about creating novel treatment protocols that are likely to be much more effective against some of these deadly diseases.
Wonderful. No, this is, you know, I really love how this, this futuristic concepts really bring the kind of stuff that engineers like me do with computer scientists, with chemists and biologists. When we say it takes a village, I can see it in a slide like this. So I want to thank you, Maurizio. We have a few minutes for questions, and I want to ask people to either raise their hand or directly in the chat box be able to post their questions. And I've seen people who've done both. So maybe I'll start off with a question in the chat box. Do you think the enormous costs associated with clinical trials hamper innovation in the biomedical sector? And what's the approach towards dealing with the risk associated with such trials?
Yeah, well, you know, there are examples of alternative regulatory processes around the world that have attempted to sort of streamline the process. You know, it's been well known that, for example, the European Medicine Agency has a different approach to that risk, and therefore there is some wisdom in doing clinical trials in other parts of the world and taking a lot of these sort of innovation from a global perspective rather than simply looking at it as a classic sort of US-based approach. That said, I think you're asking a more fundamental question, which is, can we reduce the number of failures by the time you get to the clinic that will essentially de-risk that? Can we get to the clinic so that we're pretty sure things will work, both from a toxicity and sort of risk factor perspective? And I think the prior slide, or the slide that may be still up, is your answer, right? If we had a mechanistic method to really sort of whittle down, let's talk about drugs since that's what the slide is about, but we can also do the same in diagnostic and other devices. You know, right now, the way a lot of these devices are built, a lot of these drugs are built, honestly, it's the scientific equivalent of the old conceptual exercises that if you put some monkeys typing at random on a typewriter, eventually, statistically, if you give them enough time, they'll type a novel, okay? Yes, it's possible statistically, but when you actually look at the timelines involved, they're very long, okay? Well, we're kind of building drugs the same way nowadays. Very rarely do we have a mechanistic understanding of the perfect drug. What we do is we take molecules that have a profile and for which we have data, and we kind of filter things down to those candidates that we think might work. And then we try them out, and we try them out at random. And first we try them out on animals to try to make sure they don't kill anybody. And then we try them out on patients to see if they actually work. And I'm being a little oversimplifying here, and I don't want in any way denigrate the great science that goes into trying to improve that sort of combinatorial and other sort of approaches to drug design. But it is a pretty inefficient process. And that leads us to the average drug costing $1 billion. It doesn't cost $1 billion to do all those trials. But when you look at the chances of success of all of your trials, then the ones that are successful that incorporate the cost of all the failures end up being $1 billion. So if we had a system like the one I described before, which, let me remind you, hinges on doing that sort of trial and validation, you're still doing it. You're just not doing it in a clinical setting with patients. You're doing it with a chip. And maybe patient-derived tissues or cell lines, but if you could do it that way, then you're going to dramatically de-risk. And by the time you go to the clinic, you're not only going to have a much lower chance of failure, but you're probably going to be able to do a much lower scope and size of your clinical trials. So to us, this is a scientific solution to the question. And I think it's why, if you ask most pharma companies today, I would say the impetus to go into sort of digital discovery systems with the technology such as drug on a chip is very, very high.
Wonderful. Makes sense. We have a question from Saeed Ferha. So Saeed, you're unmuted. Please go ahead.
Hello.
Hi.
Hello, everyone. Hello, everyone. Hello, Professor Padamosenia and Dr. Maurizio. Thank you for your presentation. It was so unique and different matter for the time. I have two questions. One of them is from you, Professor Padamosenia, and another one is from your guest. The question from you is, I want to know what is your opinion about chimeric embryo technology? You know, it is one of another radical matters in organ generation. How do you compare it with 3D printing in aspect of efficacy and ethical side? Because didn't you think about combination between these two?
Yeah, so definitely always on the side of being more conservative when it comes to scientific progress, when it comes really touchy subjects like that. So I believe that probably a more feasible approach right now for getting basically approaches that are acceptable by the population is through engineering approaches that I've been talking about, like materials that induce regeneration or biofabrication approaches. So yeah, but that's just a personal preference.
Okay.
And what was your second question?
Okay, because it was my big question, and I want to ask it from you later. Okay, my another question is, we know the world after pandemic would be different in many aspects. What do you think, Dr. Maurizio, what do you think about improved scientific collaborations and make traditional regulatory stages from hypothesis to clinical use more feasible for a situation like this?
So you're completely right that I think the post-COVID world will introduce some systemic changes, because I think in many levels, society is not going to tolerate, people are not going to tolerate the level of lack of preparation and lack of collaboration between the political world, in many ways, the clinical world and the scientific world. As I mentioned to you, the scientific world knew that a pandemic was coming and it was just a matter of time. The warnings from the scientific world were promptly ignored. I'm not blaming anyone in particular, but you know, often preparation, it's a little bit like buying insurance, right? It's a small investment, yet there's a lot of uninsured people. And so we as a country, we as an international community, were caught unprepared. But even branches of society, like just think about all the realities of returning to work. And you know, Amazon was already hit with 6 class action lawsuits because employees caught COVID while working in a warehouse. And ironically, they're not blaming Amazon for having caught COVID. They're blaming Amazon for having been negligent in running a population health that offers screening and minimizes the risk to the employees. So these issues, so right there is an example of the kind of things, you know, if you go into a hospital, there's infection control. And infection control has epidemiological studies, and they track whether an infection occurs, they trace it back, they do remedies, they isolate the patients, and so on. I think those are going to be the standards that you'll expect of any large employer, and well beyond COVID, okay? So the penalty there is regulatory, it's legal. You know, you might, if you don't do it, you are not just violating best practices, but you might actually be liable. Now that's a US, liability tends to be a bit of a US thing, but you know, I'm seeing trends throughout the world where sort of the awareness around the importance of applying scientific methods is to sort of maintaining safety and providing healthcare preventions is going to become the norm. And institutions and communities that normally wouldn't worry about these things suddenly will do so. Now, on the flip side, you saw the result of the FDA accelerating all sort of emergency use application. And that was a bit of a dual sword, right? Those of us that have actually looked at the performance of some of these diagnostic tests know that they've been approved or cleared, but they are all over the map in performance. And believe me, if I had to take a COVID test right now, there's definitely some tests I wouldn't take. Okay, not because they're dangerous, but they may be ineffective or marginally effective. Okay, so relaxing the regulatory process has also unintended consequences. And while it's great that we rushed so many products onto the market, it's unclear whether those products are right. But I think the biggest enduring effect, which really I think speaks to your question, is I think the connection between population health and health affecting every one of us, not just sick people with a specific disease, and the scientific world needs to dramatically improve. When we cannot communicate the risk factors, when we cannot communicate the cause and effect consequences as scientists, we then have the issue that the population doesn't understand. And if they don't understand, they might reject. And that's true of the politicians as well as the ordinary citizens. So I think there's a higher call to action where education and scientific education specifically, and communication of what are the risks and the risk factors, as well as the possibilities, needs to step up dramatically. And I think it's an opportunity for any of us in the scientific community to really step that up, because it will bring greater support, it will bring greater understanding, it will bring greater partnership between institutions and scientific institutions.
Wonderful. I'm sure there's a lot more questions. I just know that we're kind of running against time. So I want to just thank Maurizio. Maurizio and I will do many more of activities together. I do encourage everyone to reach out to us and ask any additional questions. I know we didn't get to some specific questions right now, but I think this is a beginning of a very wonderful journey that we're all taking as an institute together. So with that said, I want to thank you all for coming and look forward to our next Fireside chat. Thank you very much, everyone.
Thank you.
Thank you, Maurizio. Yep.